Right, today we are diving into one of nature's most incredible operations. We're talking about a biological heist, where a tiny bacterium breaks into a plant's fortress and literally rewrites its genetic code. It's this amazing story of natural espionage that, it turns out, completely changed science as we know it.
So first, let's just set the scene. Imagine you're a scientist, or, well, in this case, a bacterium, and you want to get a new piece of DNA into a plant. How in the world do you actually do it? Turns out, it's not nearly as easy as it sounds, mainly because plants have some pretty serious security. And this is the main problem. You see, every single plant cell is surrounded by this super rigid cell wall. Think of it like a biological fortress built out of tough cellulose. You can't just knock on the door and ask to come in. You have to find a way to breach this defense system. And honestly, that challenge stumped scientists for a long, long time.
But here's the thing. Nature had already figured it out. For centuries, people had seen these weird tumor-like growths on trees and shrubs. They're called crown galls. And for the longest time, nobody knew what caused them. This disease was actually the clue, kind of like the calling card left at the scene of the crime.
It turns out the mastermind behind these galls is this little guy, Agrobacterium tumefaciens. It's a really common soil bacterium, and it's the microscopic genetic engineer that causes crown gall disease. It figured out how to sneak past that fortress wall way before we ever did.
What's so incredible about this is that it's not just some random infection. Agrobacterium is, for all intents and purposes, a natural genetic engineer. It has evolved this incredibly sophisticated system to transfer a piece of its own DNA right into the plant's genome. It's basically hijacking the plant's own cells to create a safe home and a permanent food source for itself. So clever.
So, how does this tiny criminal pull off such a complex heist? Well, it's got a secret weapon, a specialized piece of equipment that holds the entire blueprint for the whole operation. So let's take a look at the tool of the trade, the Plasmid.
This right here is the T plasmid, and T stands for tumor-inducing. It's a big, circular piece of DNA that's separate from the bacterium's main chromosome. You can think of it as the complete heist plan. It contains every single gene necessary to find a target, break in, and deliver its, let's call it a payload.
Now, the plasmid itself is just brilliantly organized. It's split into two absolutely critical parts. First, you've got the tDNA, which stands for transfer DNA. This is the payload, right? The package that actually gets delivered. It carries the genes that reprogram the plant cell to form a tumor, and this is the really clever part, to produce these special nutrients called opines that only the bacterium can eat. Then the second part is the virulence, or vir region. This is the heist crew and all its gear. These genes, they don't get transferred. Their job is just to manage the entire operation from start to finish.
Okay, so we've got the plan, we've got the crew. Now for the main event, the infiltration itself. And this process, it's just a stunning example of molecular coordination. It all unfolds in 5 very precise steps. The whole heist kicks off when a plant gets wounded. That wound releases these chemical compounds, things like acetylsyringone, which act like a silent alarm for the bacterium. It detects this signal and the entire system just springs into action. The Vir genes get activated and they start to precisely cut out the tDNA payload. Then they assemble this incredible structure, basically a molecular syringe, that punches right through the plant cell wall and membrane. The tDNA is then carefully shuttled into the plant cell, escorted all the way to the nucleus, and then seamlessly woven into the plant's own DNA. And just like that, heist complete.
Let's actually meet the key players in this little heist crew. You've got Vir A, that's the lookout. It's a protein sitting in the bacterial membrane that senses those alarm signals from the wounded plant. Once it's alerted, it activates Vir G, that's the mission commander. Vir G gives the go signal to the rest of the team. Then you have the Vir D1 and Vir D2 proteins. They're the safecrackers. They form an enzyme that precisely snips the tDNA right at its borders, cutting it free. The Vir B complex is the transport team, the ones who build that molecular syringe. And finally, Vir E2 is the bodyguard, a protein that wraps around the tDNA to protect it from being destroyed on its dangerous journey to the nucleus. Every single member has a very specific and very important job.
Now, this natural process is amazing all on its own, but the story doesn't stop there. Scientists saw what Agrobacterium was doing and had this absolutely brilliant idea. What if we could hire this expert thief for our own purposes? And that is exactly what they did. They took the bacterium's T plasmid and they disarmed it. They cut out the tumor-inducing genes from that tDNA payload, and they replaced them with genes for things we actually want. The heist crew, the VAR genes, they left that completely intact to do its job. So what's the result? Well, Agrobacterium still performs its flawless infiltration, but instead of causing a disease, it delivers a beneficial gene. We basically turned a natural pathogen into one of the most powerful tools in all of genetic engineering.
So by understanding and then kind of co-opting this natural mechanism, we have fundamentally changed our ability to interact with the entire plant kingdom. This one little bacterium gave us a master key, and it transformed the abstract idea of gene manipulation into a practical, revolutionary technology.
So, you might be asking, why does any of this matter? Well, the impact is all around us. The very same technique that causes tumors in the wild is now used to create crops that can defend themselves against pests. It lets us engineer staple foods, like rice, to produce their own vitamins, which helps fight malnutrition around the world. It can even turn plants into living factories that produce complex medicines and vaccines. What started as a simple plant disease has become a revolutionary tool for our health.
You know, the story of Agrobacterium is just a perfect example of how studying a natural process, even a disease, can lead to these incredible, groundbreaking discoveries. A biological heist, perfected over millions of years, became our go-to tool for improving pretty much everything from agriculture to medicine. And it really makes you wonder, right? What other natural secrets are just sitting out there waiting for us to understand and maybe even harness them?